Okay, welcome to the second of these, what we might call the director's commentaries on the biochemistry tutorials, but we probably won't call them that. A little mention of the fact, and I've mentioned this in the lectures, the tutorials don't necessarily map onto individual lectures or even the order in which we did the topics in the lecture. If you do a tutorial and there's something we haven't covered yet, that's okay. Just put it one side and come back a bit later on and have a look at the tutorials. The idea of these is that you should have a look at them a number of times throughout the year. Obviously focusing in much more when we approach revision time. Okay, I'll just open up the tutorial in a second. I should mention first, somebody asked me what I use to make these tutorials. Made with a tool, something called ZertiWeb Toolkits. A free toolkit developed by the University of Nottingham, which allows us to do lots of interactivity within toolkits, within tutorials, and then you can download the tutorials, you can put them on the web, you can send them to other people. So quite a useful way of developing interactive tutorials. I might do a video or two on this later on in the year if I have time. Probably not going to do anything in biochemistry about this, but I may ask some students in other modules to maybe think about developing some Xerti tutorials. So the food students have been warned. Okay, so we're on tutorial 2. So I'm going to start looking at the tutorial. Depending on how long it takes, I might break this up into 2 parts. So I'm going to right click on there to open a new window. It happens almost immediately. It'll jump to do itself, and there's the first page. Remember the navigation tools at the top, obviously forward and back, or if you want to go to a specific area, you can just click on there. At the moment, we're just going to go through the tutorial sequentially. Okay, so here's a question. Write a short definition of a chemical reaction. 50 words maximum. Now have a little think about that. What is a chemical reaction? A chemical reaction is going to transform one set of chemicals, be it one or many, into a different set of chemicals, being one or many. But how is that going to happen? I mean, I might say, well, we're going to heat it up. Well, okay, that's the conditions. But when I say how is it going to happen, I'm thinking a bit more fundamentally than that. What we're going to be doing is rearranging the atoms in the substances. We're going to be breaking chemical bonds, and we're going to be forming chemical bonds. Often not all of them. Sometimes, especially in organic chemistry, the reaction only involves something called a functional group, the part of the molecule that does the actual chemistry. But anyway, let's have a look at what definition I've got here. So click on check. And it says a chemical reaction is a process involving the transformation of one set of chemical substances into another set of chemical substances, involving the breaking and forming of chemical bonds. An important point down here, conservation of matter, really. We start off with X carbon atoms, for example, and Y hydrogen atoms. We do the reaction, at the end of the reaction we still have X carbon atoms and Y hydrogen atoms, though we've rearranged them somewhat. Okay, moving on. Right, here's a schematic for a chemical reaction. It's got stuff over here which are the chemicals which we're going to transform into different chemicals. So A and B are going to react together to make C. And we know they're going in that direction because that's what the direction of the arrow says. I've also got 100 degrees here. So what we're going to do here is take these little sort of labels and move them across. Now starting with products, that's what we're going to end up with. So if we drop that on C, C is our product. It points out here there may be one or many products, obviously depending on the reaction. In this case, we're taking A and B, 2 reactants, and making a single product. So we're doing, in biological reactions, a synthesis reaction. In biological terms, a synthesis reaction. So I'll click on reactants there. And it does point out that in biochemistry we often refer to reactants as substrates. And that's probably the word you should start using from now on. If you look at information about metabolic pathways, they will talk about the substrates of the pathway. That is the individual reactants. So substrate A is converted to substrate B, C, D, E, etc., etc. Okay, the direction of the reaction, in this case, is said to be unidirectional. It's going from A and B to C. Normally we place the stuff that's going to change on the reactants are placed on the, that's the wrong way around, isn't it? Yeah, that should be the reactants are placed on the left hand side and the products are placed on the right hand side. I guess this derives from Western ways of writing. We write from right to left, left to right rather, and down the page. Okay, moving on. Okay, so here's a schematic for a biological reaction. Again, we've got our substrates, or reactants if you will. We've got our products, in this case C. We've got our conditions. In this case, we've said we need the molecule adenosine triphosphate, which I have covered the structure of that in other videos, and I think we've got a picture of it a bit later on. That is the conditions. It's a bit of a simplification of the conditions there, actually, because we'd probably, if there's a biological reaction, we'd also need an enzyme. It would have to be in aqueous conditions, probably at body temperatures of a human being. You probably need some magnesium associated with the ATP. Generally speaking, when you draw reactions, you summarise the conditions above the direction arrows. But now note this arrow is a bit different than the one we saw earlier on. That was a unidirectional one going from A and B to C. This is much more like a typical biological reaction. Under certain conditions, and using a particular enzyme is probably the most important condition, we convert A and B to C. Under other conditions, we can convert C back to A and B, almost certainly using a different enzyme. So that's why we've got this funny little pair of half arrows, indicating the reaction is a reversible reaction. It can go in either direction depending on the conditions. In biology, as I say, conditions typically will depend on which enzyme is active, but that will also depend on how much we've got of these things. So for example, if we don't have much of C and our body needs C, the reaction will tend to go in that direction from A and B. Conversely, if we have a lot of C and we can think of, our body can think of some other uses for A and B, we're liable to take C and break it down to make A and B. It's quite a short biological pathway here. We go from A to B, reversibly to C, and then possibly to D, then possibly to E, and there's possibly side reactions coming off as well. And this is the nature of biological reactions. They are both reversible and they are deeply convoluted, so lots of things can be happening simultaneously. So the forward reaction A and B to C can be happening at the same time as the reverse reaction is, though almost certainly at a different rate. Okay, energy in chemical reactions. I've mentioned a few times that chemistry tries to get the lowest energy configuration, and this is the conventional way of drawing reaction progress. It doesn't look like much at the moment. We've got a couple of axes. We've got a line that's going up and down, and we've got a dotted line. Now what this does is allow us to model change in energy in the reaction. So first off, we'll put in some labels for the axes. And I'm going to put in reaction progress first. So it's on the x-axis and it tells us how far the reaction has progressed from substrates to making products. So over here it hasn't progressed at all. Over here it's completely finished. And over here we have this funny little halfway state, which I'm going to explain in a little bit. On the y-axis we put energy. So drop energy in there. The reaction progress you can measure in, do we have substrates, do we have products? Okay, do we have a unit for that? Probably not. For energy we do have units. Specifically kilojoules per mole. So that's a measure of both energy, kilojoules, and mole, of course, is a measure of quantity in chemistry. So kilojoules per mole, you probably have this label as an axis. Right, we know we started from substrates, so if we drop substrate in here, that's a reasonable place to start. That refers to the amount of chemical energy possessed by the substrates at the start of the reaction. Now this is actually quite a complicated concept. What we do is often measure the heat change, the changing the amount of heat in the system between here and the reactants, and the products rather. There are quite a lot of other different things happening as well, which I will talk about a bit later on when we talk about free energy. Okay, so the products come over here. So this is the amount of chemical energy possessed by the products at the end of the reaction. A few mistakes here. That should be end of the reaction. Okay, we start from the substrate, which has quite a high amount of energy. And if we ignore the middle bit for now, we end up with a product which has a lower amount of energy. And that's what chemistry likes to do. It likes to go towards the lowest amount of energy. There's a bit in the middle, and luckily we've only got one label left, so we'll just drag that over and drop it in here. This is the transition state. All reactions go through a transition state. If the difference in energy between here and here is very high, the reaction will be very difficult to make go. It will be very difficult to make the reaction happen. Now what happens in biological reactions is that typically if you just try to convert the substrate, you mix it up in water with some other chemicals and try to make the product, it won't happen because the transition state is at such a high energy level. What happens is molecules, mostly proteins, a few RNAs called enzymes, reduce the magnitude of this transition state. I haven't drawn it on this diagram, but effectively that point will be lowered down to somewhere here, making the reaction much more easy to happen. Enzymes are said to mediate biological reactions. That is, they reduce the value of the amount of energy needed for the transition state, so facilitating the production of the product from its substrates. There may be a number of different transition states, it has to be said, in complicated reactions which take a number of multiple steps. And then, of course, each individual reaction is probably involved in quite a lengthy pathway. Okay, we're going to have a look at some important classes of biological reaction. There are many, many hundreds of thousands probably of different biological reactions. Really only a few, actually about 6 different main classes of reaction. If you can understand what these are, you'll be fine in developing your understanding of biological reactions. So this is one of these things where you've got to have done a little bit of reading, and then you type in the single word in lowercase, and it gives you, tells you whether you got the answer right or not. So a class of reaction that involves the formation of large molecules from small molecules, often involving the loss of water. So this is, oops, I don't know if I can spell. Synthesis, we hope. And it is synthesis. Now the opposite process, where we take big molecules and we break them down, often involves addition of water, which is a bit of a clue because the general name for this class of reactions is hydrolysis. Again, we're right. Okay, now here's something we need to read a bit carefully. This class of reactions can be described in a number of ways: gaining oxygen or loss of hydrogen, or loss of electrons. The simplest way is probably gaining of oxygen, so that should be suggesting to you it is an oxidation reaction. And the next one is the opposite case, where they lose oxygen or we gain hydrogen. Or we gain electrons. In this case we would call that a reduction reaction. And it's right, so we did well there. So moving on. And here's an example. Here's an example of a synthesis reaction. We said it involved loss of water. Synthesis reactions typically to form bonds. So if you look at the two ends of these two amino acids, which is what they are, this one has an amine group there with 2 hydrogens attached to it, and this one has a carboxylic acid group attached to it, again with an oxygen and an OH group there. Now if you look what happens to this carbon here, it becomes this one here, but it's lost its OH, so remember that OH, and have a look at what happens to this nitrogen, which has 2 hydrogens associated with it. It becomes this one, with a bond formed between it and the carbon. So overall this carbon has lost an OH, and this nitrogen has lost a H, and that's where the water comes from. Okay, here's the opposite case. It's a bit hard to see, the diagram's not particularly good here. This is effectively a long chain sugar, a polysaccharide of some sort, a bunch of glucose molecules tied together. And if we chop it up, for example, we take potatoes and we boil them up in a pan. If you try to eat potatoes raw, you'll feel very sorry for yourself reasonably soon afterwards, because we can't digest the starch molecules are too big. But when we heat it up, we hydrolyse them by adding water across this bond here. So there's single oxygen there, and if you look down here, it's gone to a HO plus another hydrogen, so we've actually added water across this bond, and that forms what's known as a hydrolysis reaction. When you heat stuff up, it does it quite effectively. It breaks starch down. Enzymatically, it's quite a little bit harder, which is why your body isn't equipped to break down the long starch molecules in uncooked potatoes, for example. What typically happens is an enzyme will start at the end of a polysaccharide, so it's got to work at the bit of the end, then it chops a bit off, then it's got to chop the next bit off, having shortened it a little bit, and so on and so forth. Whereas when you heat stuff up or use other methods such as acid hydrolysis, it breaks stuff up in a chemical fashion. It does so much more efficiently, I guess, at least from that perspective. Okay, we'll probably have a break after this one to introduce the most famous hydrolysis, that of ATP. Right, you need to remember what ATP stands for. There's a number of possible examples there, and if I put in adenosine trisphosphate, which sounds sort of good. No, close but not quite correct. Adenosine 3-phosphate, I would hope nobody would put that one in. Yeah, it's not quite correct. So the one we want is adenosine triphosphate. Oh, well, trisphosphate. I put trisphosphate in again. Oops. That one, correct. We got there in the end. This is the molecule. Anyone who's seen my series of on Accelerist draw, which I actually drew this in Accelerist draw, will know what these various parts of the molecule are. We have the adenine nucleotide base over here. It's nucleotide base. We have a ribose sugar here, and we have 3 phosphate groups. Okay, there's a little bit of important reading about phosphate groups there, so you may want to pause the video and have a read about them. Okay, this is hydrolysis of ATP. ATP plus water makes adenosine triphosphate plus water makes adenosine diphosphate plus free inorganic phosphate, which is often, for example, incorporated into another molecule. So the question here is, we've got ADP, we can hydrolyze this further, so what we're going to produce. You don't need to know anything about the structures, you just need to think about the basics of how ATP is put together, and once it's lost a phosphate, what will it look like? So have a little think about that. Oops, went a bit far there. And the answer is this, adenosine diphosphate. Again, we add water to perform the hydrolysis, and we end up with adenosine monophosphate. So instead of having 3 phosphate groups in the tri, we went to 2 in the di, and we now have 1 in adenosine monophosphate. So here's an example of this reaction. This is the first example reaction in a process called glycolysis. It's a bit hard to see what's happening here. The graphics aren't particularly great, but here's a molecule glycolysis. There's an enzyme, hexokinase, and magnesium, and some ATP, which gets hydrolyzed to ADP. And what happens to the phosphate from here? If you look closely, it's replaced that hydrogen there. This is quite an important biological reaction, and you need AP to make this happen for a couple of reasons. Obviously, somewhere to get the phosphate from, but also, as we'll discover in later lectures, because it provides energy for the reaction to take place. Actually, I think I'm going to probably continue. It's getting a bit lengthy now, but no, I won't. I'll stop. It's getting up to 20 minutes. So I'll pause now, and I'll come back with page 15 shortly.